Bismillah Rahman Rahim. Welcome back, friends. Welcome to another video lecture from Kami Biology. So we talked about the mechanism of action of this sulfonamide, or we can say folic acid inhibitor drugs. Now this is the part B of this video of this sulfonamide videos lecture, and in this part B we are going to talk about resistance which are activated against these drugs. So let me explain. Resistance against metabolic pathway inhibitor or folate synthesis inhibitor, or we can say sulfonamide. Now if we talk the resistance, so the bacteria, if we see in mechanism of action that the drugs will perform their mechanism of action, so then bacteria will also produce a kind of resistance. So there will be many resistance. When I prepare this one topic, so I saw many of the resistance, but it will confuse you. So what actually I am going to talk? I am going to talk about 2 or 3 way of the resistance. The first way of the resistance, all of you better know about the efflux pump. So I already explained it in the 3 or 4 videos of this antibiotic. So if you miss these efflux pumps, so then go back and watch the video. So the first way is the production of efflux pumps. Due to this efflux pump, the drugs will get internalized through porine channel and then will again release out by this efflux pumps. These are the actually holes. Now the second way of the resistance, if we talked about the second way is the mutation, and we usually call that spontaneous mutation. What actually happens in spontaneous mutation, the structure of these enzymes will be changed. Let me erase this one area. So the structure of enzymes will be changed. Due to spontaneous mutation, there will be a resistance genes which are present inside a bacterial cell, or the plasmid will produce a kind of resistance genes. Due to this resistance genes will be shifted to the bacterium genome, and then this bacterial genome will produce an enzyme, and we call it mutated enzyme, or we call it mutated dihydropetroate synthase. So this is actually mutated dihydropetroate synthase. So how these enzymes will be looked like? So if we see the structure of the enzymes, so these enzymes will be means a slightly modification, or we can say a slightly alteration will be occur at the active site of the enzyme. So due to this slightly modification, let's suppose this is the drugs. The drugs will not bind. The drugs will not bind. They will lose their affinity. But the substrate will bind, means PABA and dihydropetroate diphosphate will be bind here. So let's suppose these are the substrate. The substrate will easily bind here due to this active site. But this drugs, mean this sulfonamide, will lose their affinity due to this alteration of this enzyme. So when the enzymes will be altered or mutated, so the drugs will be not bind. So due to this way, again the products will be formed, or we call this product, as you better know, means dihydropetroic acid. So what we get? We get the dihydropetroic acid. So this is the product. So then finally we get the product. So this is the first way of the resistance which are activated against these drugs, means mutation. Now the second way of the resistance, or we can say a third way of the resistance, if we talk, so this is the high concentration. High concentration of what? High concentration of substrates. What actually happen? The PABA, if we see the structure of the PABA, so the PABA will be a high in number. So the PABA will be in a high number. So the PABA produce in a high number, as well as the substrates, another substrate we also call that dihydropetroic diphosphate. So let me write in a short form. So due to the high production of these two substrate, means these two substrates will be in a high production. So due to these two substrate in a high production, we can say they will be produced in a high production. So the drug's production, as in this case, will be low. So the drug's production in this case will be low. Why? Because there is a high number of this substrate, mean this PABA and this dihydropetroic diphosphate. So what actually happened in second case, let me once again, the bacteria produce these substrate in a very high number, in a very high amount or high concentration of these substrate will be occur. Due to this way, this drug's concentration will be loosed, or we can say the concentration of these drugs will be low in number. Why? Because they are high and they will be automatically in low amount. So due to this way, these substrate will be easily bound with the active site of the enzyme, but they are not bound. Why? Because there is a low amount of this substrate as well. So due to this low amount or due to this low concentration, these substrate will be easily bound, but the drugs will be not bound. So this is the second way of the resistance as well. So due to this resistance, again the whole process will be continued as I explained in the previous video lecture. So again folate synthesis will be started, and again purine and pyrimidine will be occur. So that's all about the resistance which are activated against these drugs. Now let's talk about the side effect which are activated against these drugs. As you better know that when again we are giving these drugs, as we know that these drugs will create what? These drugs will create a resistance, or the resistance the resistance is activated against these drugs. So there will be a side effect when we again used these drugs again and again. So which kind of the side effect? Let me explain. High V's. The first side effect is the high V's. In this case, means red H's will be a pair which is very itchy, or we can say these are the very irritated one. This is the first way. The second way is the fever. Rashes. Fine. So these all about the side effect which are activated against these drugs. Although there are some other, but these are the exact. So I hope all of you get the idea about the metabolic inhibitor drugs, the side effect, mechanism of action, and the discussion about sulfonamide, their structure, and everything about these drugs. So I hope I explained it in a very in a very detail. But still, if you have some questions or still if you have some confusion about that topic, so write it into a comment. Inshallah, I will try my best to give you answer as soon as possible. So if you like this video, please hit the like button, share this video, and don't forget to subscribe the channel. Subscribe the channel because in the next video we are going to talk about some other antibiotic drugs as well. Thank you so much.